DNA repair is a key factor in both causes of cancer and in the treatment of cancer. So this is an ideal time to have a special conference on DNA repair. One of the most exciting parts of this AACR meeting has been looking at how cancers are often defective in their ability to repair DNA damage. And this presents an Achilles heel for a whole new spectrum of treatments.
The important thing about this meeting is that it is going to be talking about the clinical endpoint to a lot of the basic biology of DNA repair. So we certainly want clinicians to come to this meeting, residents, fellows who are interested in this type of area of research, but also, of course, the basic scientists who want to translate it and actually impact on the clinic. We want them all. A smaller number of people at a conference like this really allows interaction with young researchers. And actually, of course, they are the future for cancer research, and it adds momentum to our field when we can get young researchers engaged right off the bat with the best in the field. And I think that's exactly what this meeting is going to do. And also, of course, we'll be looking at novel data that will not come out in publication for 6 to 12 months, perhaps. That's important too, because it gives you one step ahead in your own research to understand if something is coming up that might actually affect your own research, you can actually get on top of that as well.
One of the best features of these kinds of special meetings is the ability to expose young investigators to the field as it's developing. So to put a young investigator side by side with the senior person, so the senior person can both describe the field and describe where the field is going, but at the same time can communicate a passion for where that field is going to the junior investigator. It's great for a junior investigator to read the literature, but you can read hundreds of papers and still not get that big picture. When you go to a meeting like this and the senior people can lay out where the field has been, what are the big questions, that in a couple of days, a junior investigator can pick up the entire field, know its history, know its direction. It's the kind of thing that you would take 1,000 hours of reading can be accomplished in just a few days of a special meeting like this.
The topics of this meeting really are going to be about the fundamental processes of how cells repair DNA damage. So we're going to be looking at different repair pathways. This includes homologous recombination and non-homologous end joining of double-strand breaks. It's going to include looking at the DNA damage response and cell cycle checkpoints as well. And importantly, it's also going to look at the huge amount of data now that we have from large sequencing studies as part of the TCGA and ICGC, and really put it together, which is, one, what really are the targets that we should be looking at that have faulty DNA repair in terms of the types of cancers that we should be targeting with novel therapeutics. And two, if you actually have abnormal genomics within cancers, in fact, what are the deficient repair pathways that led to that? And again, is that an Achilles heel as we go forward? So we'll have panel discussions after the basic science sessions to actually ask about the clinical applicability. Can we use these as biomarkers, for example, in clinical trials? Can we drive forward novel drug treatments, and can we use this information with radiotherapy and chemotherapy that currently exists? And that panel session I'm really looking forward to, because that'll be a mix of basic scientists and clinicians trying to work together and go forward.